Alrighty guys, and now on to section 2 with our notes. So we are starting here with energy and chemical reactions. Now a chemical reaction is represented by some type of an equation. From our vocab terms, if you watched that video, we know that the reactants are going to be on the left side of the arrow, the products are going to be on the right side of that arrow. So here I have a very simple reaction. I have 2 hydrogens plus 2 oxygens, and that is going to give us H2O. You're probably wondering, well, that doesn't seem balanced. Where is the other O at? And that other O was actually given off. He's no longer a part of the water molecule. Metabolism. Back from chapter 1, we talked about how the metabolism is the sum of all these chemical reactions in your body. Now your body is undergoing reactions continuously, and that is allowing you to have energy so that you can function, so that you can sit here at the computer and watch this video, so that you can go outside and take the trash out or get the mail. For example, the rock here, he has a lot of reactions going on in his body right now because he's working out. I used to think he was really big in that picture, and then something happened and now he's like the size of a bus. I don't know what he did, but chemical reactions are happening all of the time within yourself. Most chemical reactions are going to require some type of energy in order to begin, and that energy is called the activation energy. So the goal of this little guy here, he is trying to make this boulder roll down the hill. In order for it to roll down the hill, he has to push it up the hill first. So this would be considered the activation energy because it's not going to roll down the hill until he gets it to the top. So he is pushing it up the hill. Once he gets to the top, the reaction will begin and it will go straight down, landing at the bottom. Just another example here, you can see the reactants have to go up that steep hill first, and then they come back down the other side. And that's where they turn into the product. Now if we add a catalyst into this reaction, remember a catalyst is going to decrease that amount of activation energy. So it's going to make the reaction happen sooner, and if this is happening inside of an organism, they're not going to need to use as much energy. So there's a comparison between the two. If you can use a catalyst, you want to use a catalyst. So down here, you can see that there's the reactants. We have red ones and blue ones, and the activation energy is taking them up to the top. Once we get up here, now the reaction is going to begin to occur. So these guys are going to become combined, and they're going to come down the other side as the products. So what started as 2 things over here is now 1 product at the end. Okay, here's another example with a catalyst. We know that catalysts, again, reduce the amount of activation energy. So here's the same reaction. The left picture, reaction 1, is without a catalyst. So if you look at this, you can see there's a large amount of activation energy needed. If you look at reaction 2, this is the one with a catalyst, and there's not as much activation energy needed for that one because we were able to use that catalyst. Inside of living things, we're going to call a catalyst an enzyme. You actually have enzymes inside of you that help your reactions to take place. And some people will actually take enzymes in order to help themselves out. You can give them to animals as well. You can sprinkle it on their food, and you don't really notice that you're eating it, or the dog won't notice that it's eating it, but your body, or its body, will take those enzymes and use them within reactions. Again, just another picture of a catalyst versus non-catalyst. So if you're looking over here at this one, normal activation energy. This one down here, it's lowered by, I'm calling it an enzyme down here. We know that the enzyme is a catalyst. However, this reaction must be taking place within a living organism because we're saying it's an enzyme. Now in class, normally I would have you guys go through, you would draw this diagram out, and we would label the parts. But since we can't do that, I am just going to tell you them. Over here, we have the reactants. This is where they are starting at 50. Take note of that. Starts at 50, not at 0 in this particular graph. Here's our activation energy, so it's going from the 50 line all the way up to the 250 line. Now, normally I would ask you a question like, how much activation energy is needed? People always want to go straight up to the top here. Oh, 250, that's how much energy is needed. No, that's not actually correct. Remember, we started down here at 50. So you would take 250 minus the 50, because that's where the reaction started, which would be 200. So you would need 200 parts of energy in order to make the reaction begin. Kilojoules, we could call it. And then your products are ending over here. Exothermic. I'm going to throw a couple other words out at you here that I did not include in the vocab. Exothermic is a way we can describe a graph if heat is being given off. So here we have our reactants, here's our activation energy, and then the products are below the reactants. So this would be considered exothermic because it actually gave off more energy, or it gave off more heat in order to lower itself below where the reactants were. So if your products are below reactants, exo. On the opposite end of that, if the reaction is taking in heat, then it's going to be endothermic. I always thought the prefix endo kind of sounded like into, and that's how I remembered something was coming in. Exo was when things were leaving. So if heat is absorbed, you'll notice that the products are above the reactants. Now looking at these two graphs here, we can see we start with the reactants, get our activation energy. This graph down here, our products are above our reactants. So because it is above, this reaction took in heat. This is considered an endothermic reaction. This reaction over here, the products are below the reactants. That means heat was given off, so that is going to be called an exothermic reaction. Enzymes. Enzymes are proteins that act as catalysts in living organisms, and each enzyme can act on a different type of a substance. So there are specific substances for each enzyme, and you have hundreds of different enzymes inside of your body. It kind of works like a lock and key, or think of it like a puzzle piece. Now, enzymes can mold ever so slightly to better fit their substrate, but you cannot fit a shape like this into one where a circle is supposed to be. It's just not going to fit, and that enzyme will not work on that substrate if it doesn't fit. There will be no bonding, and it'll just leave. I'm going to show you a quick little video on enzymes. Again, you may need to adjust your volume settings so that you can hear it, and then we'll finish up our notes.
Hey, Professor. Just the other day, I saw this small man swallow an entire steak bigger than the size of his head. And it got me to thinking, how could he possibly digest all that in just a few hours?
Well, the steak is first chewed in the mouth into bits and pieces that fall into the stomach. You might already know that the stomach contains a powerful acid that can dissolve even metals. But what you did not know is that this acid is not what ultimately breaks down the steak. No, it's very tiny and special molecules called enzymes that do the job. Strangely, these enzymes find the stomach acid a cozy place to work in.
Whoa! It's like small piranhas finishing up a whale in a sea of acid. Imagine what I could do with that.
Ooh. Huh?
Well, it doesn't work that way.
What do you mean it doesn't work that way?
I knew you were going to say that. To find out, you need to know what exactly enzymes are and how they work. Whenever you're ready, click Next to continue. What are enzymes? They are proteins that speed up chemical reactions. Without them, reactions can still occur, but they would be too slow to support life. There are many different types of enzymes, and they come in all manner of shapes and sizes. For example, what you call the piranha enzyme is actually pepsin. It is only found in the stomach and digests proteins. In your mouth there is amylase, which breaks down starch. In your cells alone are hundreds of enzymes that control your cell activity and defend them from invasions by microbes like bacteria and viruses. Though enzymes can appear very different, they all share a few characteristics in common. Let's now take a look at what these are. Number 1. Each enzyme has an active site. This is the groove in the enzyme where the molecules they act on, called substrates, are captured and made to undergo a reaction. The substrates are either broken apart or combined together to form products. For instance, starch found in the bread that you eat is digested to form simple sugars called glucose that is then absorbed into the body. Glucose is used up as energy, but when there's too much of it, special enzymes combine the glucose molecules together to form a giant molecule called glycogen, which is then stored away in the liver and muscles for later use. Number 2, enzymes are very specific. Each enzyme can only bind to one type of substrate. Let's look at the case of lipase enzyme, which breaks down fats. Molecules like glucose and proteins cannot fit into the active site of lipase. Only fat cells can bind to the active site because they have the right shape. This ability to act on just fat cells is very important for lipase. Lipase converts fats into energy. Now imagine what would happen if lipase can also convert proteins into neurochemicals for the brain to function well. If you no longer need the extra energy from your storage of fats, lipase will be deactivated. But this means that lipase can no longer produce precious neurochemicals for your brain. In other words, when you stop burning fats, you end up damaging your brain. To prevent such freakish scenarios from happening in our body, enzymes must only act on one type of molecule. Number 3, enzymes are recycled. Enzymes are not altered by the reactions they speed up. Immediately after they finish one reaction, they can act on a new substrate. This way, only a small amount of enzyme is needed to speed up thousands of reactions.
Alright, so that is the end of that video there. Make sure that stops. Okay, let's get back to our notes then. So, as you noticed in that video, it did talk about how you had to have a specific enzyme for each substrate. And if it doesn't bond, then no reaction will occur. That enzyme will not work. So the specific name for the substance that an enzyme works on is called a substrate. And here I'm showing you a video of what happens as far as enzymes connecting to the substrates. They fit directly into the active site of the enzyme. Enzymes are reusable, so after it's used once, that substrate will then leave, and that enzyme will go out there and find a new substrate. Enzymes are always going to end in the 3 letters A, S, E. That's how you know that it is an enzyme. If it doesn't end in ASE, probably not an enzyme. Examples of these would be lactase, which is going to break down milk. Lipase is a fat-eating enzyme, and then there's also maltase. Enzymes are going to speed up your reactions by lowering the activation energy, and they're going to model a lock and key or a puzzle piece. So here you can see there's 2 different substrates coming in there. Or no, you have one substrate coming in there, it's going to bond with sucrose, that enzyme connects to it, and now it is going to split them apart. And then that enzyme is going to go out there and find another sucrose molecule to split apart. So notice that a little bit of water is also coming in there, and that is helping that reaction to separate. Denaturing enzymes. So I just said a bit ago that enzymes are reusable, and most of the time they are, unless they become denatured. This is a great test question right here. How can an enzyme become denatured? Well, there's two main reasons. It can be ruined by temperature or acidic levels. So if the temperature is off or your acidity is off, that enzyme kind of just folds in on itself and it collapses. That means that no substrates can fit in it. So this is what it should look like, and this is what happens after it gets denatured. Notice there's no active site there anymore, and that substrate cannot fit in there. Solutions. We're almost done. Solutions are made of two parts. There's a solute and a solvent. A solute is the substance that is getting dissolved. So if you are making sugar water, for example, the solute would be the sugar because you're going to be dissolving that in the water. In this example here, these little orange guys, those are the solute, and the solvent is like these little blue guys. It's representing water molecules. The solvent is the substance that is going to dissolve the solute, and they say that water is the universal solvent because almost everything can be dissolved in water. Now, you're probably automatically going towards the fat section here and thinking about oils and butter. Those things cannot be dissolved in water. Acids and bases. So acids are when the number of hydrogen ions, so that's a capital H, are greater than hydroxide ions. And I didn't get any pictures of acid and base reactions, not reactions, but burns, because they are way too gruesome to put online here, in my slides at least. Please don't look that stuff up, it will make you nauseous. Bases are when the number of hydroxide ions are greater than hydrogen, so it's the exact opposite of an acid. You may have heard of the pH scale before, and usually we do a little lab with this stuff. If we have time when we get back, we'll do it at that time. So the pH scale ranges from 0 to 14. Acids go from 0 to 6.9, with 0 being the strongest acid. Bases are from 7.1 to 14, with 14 being the strongest base. Notice that I started here with 6.9 and here with 7.1. So right in the middle there is that number 7, and that is considered neutral. And they say that water is neutral. Here's just a general pH scale. On this side over here we have acidic things. Over here is alkaline. Now alkaline is just another name for base. We like to have multiple names for things in biology. It's just something you kind of have to get used to, for lack of a better term. So battery acid is extremely acidic. And then notice right here your stomach acid. There's hydrochloric acid in your stomach. And that is super acidic as well. More acidic than lemon. We also have vinegar is acidic, and we know tomatoes are acidic. Coming on up here to the neutral things, you have milk, water, and your blood is more basic than it is acidic. So that kind of helps to balance out, your stomach acid being so acidic down there. It's also why your stomach acid needs to stay contained, and if it gets out, that can just wreak havoc on your body there. All of your cleaners are normally more basic or alkaline. So things like, bleach or any kind of dish soap or like toilet bowl cleaners, those things are all going to be very basic. Anything that's too basic can be harmful to you. Anything that's too acidic can be harmful to you. So you ideally want to stay right there in the middle with that water at neutral. And that is where we are going to stop for today. We'll pick up with section 2.3, which is the last section of chapter 2, and that's all about water. Alrighty guys, I'll talk to you later.